competitive inhibition. Right, so a competitive inhibitor is a molecule that has a close structural resemblance to the substrate, meaning structurally it resembles the substrate, it has a similar shape to the substrate, and because of that, the competitive inhibitor is also complementary to the shape of the active site of the enzyme. It can bind to the active site. So you have the substrate that wants to bind to the active site, you have the competitive inhibitor that's also able to bind to the active site. So in the presence of the competitive inhibitor, it will compete with the substrate for the binding to the active site of the enzyme. Thus the name competitive inhibitor. Right, so when the substrate concentration is low, when there are fewer substrate molecules than the inhibitor molecules, then the probability of the competitive inhibitor binding to the active site would be higher than the probability of the substrate binding. And when the competitive inhibitor binds to the active site, as long as it stays bound, then obviously the active site is blocked, right, from binding to the substrate. Right, so the formation of the enzyme inhibitor complex prevents the formation of the enzyme substrate complex. Right, so again, the likelihood, right, of the competitive inhibitor binding to the active site of the enzyme is higher when it is at low substrate concentration. Right, as mentioned just now also, right, formation of EI complex prevents formation of EX complex by blocking the active site, preventing effective coalition, and therefore this lowers the rate of reaction. Right, so when the substrate concentration increases, when there are more substrate molecules, then the likelihood of the substrate binding to the active site increases. Right, so as substrate concentration increases, there would be more effective coalition between the enzyme and the substrate, so a higher rate of formation of the EX complex and an increase in the rate of reaction. Right, so increasing likelihood again of binding of the substrate to the active site when there are more substrate molecules, right, when the substrate concentration increases. So likelihood of forming ES complex increases. Okay, so when the substrate concentration increases until a very high concentration, the substrate molecules would be able to outcompete the inhibitor, the competitive inhibitor, to bind to the enzyme. So the probability of substrate binding is similar to when the inhibitor is absent. Right? So the number, right, of substrate molecules is so high, right, so much more than the competitive inhibitors, right, that it is as if the competitive inhibitor is not there. Right? The probability of binding of the substrate is so high, right, it's almost the same as when there is no competitive inhibitor. Right, so in this case, right, we would say that the competitive inhibition is overcome by the high substrate concentration. The substrate has outcompeted the competitive inhibitor to bind to the enzyme. Right, so the same Vmax can be achieved as if there is no competitive inhibitor, but the substrate concentration at which this Vmax is achieved would be much higher. Right, so putting everything into a graph, right, this shows the effect of increasing substrate concentration on the rate of reaction with and without the competitive inhibitor. Right, the green line would be the one without the normal one that you have learned in the previous lecture. The red line is the one with the presence of the competitive inhibitor. Right, so as mentioned just now, right, when the substrate concentration is low, right, there is competition from the competitive inhibitor that prevents or slows down, right, the formation of ES complex. Right, so for the same substrate concentration, the rate of reaction is lower with the presence of competitive inhibitor. But at very high substrate concentration, the substrate outcompetes the competitive inhibitor. Right, the competitive inhibition is overcome. Right, so it can achieve the same Vmax, but the substrate concentration for which the Vmax is achieved is is much higher, right, compared to when the substrate when the competitive inhibitor is absent, as shown here, right? So this substrate concentration is required with the presence of the competitive inhibitor for Bmax versus this one, right, which is much lower without the competitive inhibitor. All right. So using the figure just now, right, spend some time to check the Km values and compare the affinity of the enzyme. We call that from what you learned last lecture. Alright, so you can pause the video, right, spend the time to find the Km and the affinity of the enzyme and compare when there is and there isn't the competitive inhibitor. Okay, you can resume the video once you're done. Alright, so now that you have found the Km and the affinity, right, you should see that the Km of the enzyme in the presence of the competitive inhibitor is higher when there is higher than when there is no competitive inhibitor, right? So affinity is given by 1 over Km. So the affinity of the enzyme with the competitive inhibitor is lower than when it is without the competitive inhibitor, which means that the effect of the competitive inhibitor is that it lowers the affinity of the enzyme, right, for its substrate. Okay, so you have looked at the mechanism, how the competitive inhibitor works. This is the result in terms of enzyme kinetics. Right? It lowers the affinity. It is harder for the substrate to bind to the enzyme because of the competition posed by the presence of the competitive inhibitor. It is harder to bind, therefore lowering the affinity of the enzyme for the substrate.